Hi guys, this is a neuro review on the limbic system. This is lecture 1 of 2 for lab 10, the final lab for neuro. And not the final video, but we'll just get to it. Hope you guys are doing well. This has been a long neuro unit. So anyways, here we go. We are going to be talking about the limbic system, and we have the limbic association cortex, which is exactly that, cortex. And you can see it here. And we'll go into more detail. We have the hippocampus, which is going to be dealing with memory formation, the amygdala, which is our aversion center, so we're going to be dealing with fear there, and some emotional processing. And then we have basal forebrain and the nucleus accumbens, and that's going to be our gratification center. So the limbic association center, or limbic association cortex, the hippocampus, the amygdala, and the basal forebrain and nucleus accumbens, which are associated with all the different parts of the limbic system. All right, so starting off with cortex, we have the limbic association cortex is going to include this orbital frontal cortex, part of the prefrontal cortex of the frontal cortex. That's a mouthful. Then we have the cingulate cortex, which we can see here. This is cortex that is above the corpus callosum, which you can see here. So again, this is just all cortex, limbic association cortex. And its purpose is going to be to integrate sensory motor information to help shape cognitive and emotional responses. All right, and then finally we have the perihippocampal gyrus right there, which continues as the entorhinal cortex. So the orbital frontal cortex, then the cingulate cortex, then the perihippocampal entorhinal cortex, and entorhinal is going to be adjacent to our hippocampus, or part of it. The hippocampus is really just like a folded cortex itself. All right, and so what is this going to do? It's going to control learning, memory, and emotions, and it is fundamental for cognitive and social interactions that allow humans to survive and adapt a complex environment. It's going to exchange information with all areas of the neocortex. Remember, neocortex is most of the cortex. It has 6 layers. And then, so it's doing information exchange between different cortex regions, and that exchange is fairly complex, but the limbic cortex is going to integrate, as we said, integrate sensory and motor information in a way that shapes cognitive and emotional responses. All right, but the limbic association cortex is the first part of our limbic system. We have the frontal orbital lobe, part of the frontal lobe, prefrontal lobe with the frontal lobe. Then we have the cingulate gyrus, which you can see sort of wraps around, and then it extends to the perihippocampal gyrus, which extends to the entorhinal cortex. All right, but controls learning, memory, emotions, exchanging information with all the different parts of the cortex overall, and then we'll see its connection in just a second via the entorhinal cortex to the hippocampus. But let's just take a quick look at our software. So here we have the rostral part of the brain. Remember, rostral to nostril, meaning closer to the nostril. So this is the frontal lobe. Here you can see we have the cingulate gyrus above our corpus callosum. And then we don't see the rest of it, but it's going to wrap around and at the bottom on the more caudal side of the corpus callosum. And let's just look at the type in limbic system. Here we go. All right, so we can start to see the limbic system. There's really not that much to it, but we have the hippocampus, and then it's going to have a projection called the fornix. This is going to be its output section. So the hippocampus, then the crus of the fornix, again, this is the projection fibers, the axons, body of the fornix, then the column of the fornix, and that's going to end in the mammillary body. And we'll talk more about that in a second. And then here you can see embedded inside of the limbic association cortex on that left-hand side, we have one of the amygdaloid bodies, aka the amygdala. All right, but just quickly, we have this association cortex that sort of is wrapped around the corpus callosum and wrapped around this hippocampus. All right, so far we're starting to get 2 parts of our limbic system: limbic association cortex and then the hippocampus. All right, and limbic association cortex, it's integrating information, talking to different parts of the neocortex, right? Most of the cerebral cortex is neocortex, 6 layers, integrating sensory and motor information to shape cognitive and emotional responses, okay? Then we started to look at the hippocampus just quickly, but it's located deep to this perihippocampal gyrus, and it kind of looked like this, weird wrapped around thing right here, and we saw that, here's the body of it, and then it had this extension coming off of it that was the fornix. but it's located deep to the parahippocampal gyrus cortex. Right, gyrus is the ridge of a cortex, and adjacent to the inferior horn of the lateral ventricle, and it's posterior to the amygdala. And remember, we saw those amygdaloid bodies right here. So we're just saying, this hippocampus is posterior to the amygdala, which we see right in front of it. We see it wraps around with the fornix, and then ends in our friend the mammillary body. Okay, so the hippocampus and its projection projection pathway, aka the fornix, is a C-shaped structure. It is actually essentially a rolled up sheet of cortex containing 3 functional areas that look like a seahorse in cross section. I guess, yeah, that's it's made up of archicortex. So we said neocortex was 6 layers. A different type of cortex is archi, which has 3 layers. We mentioned that in the previous lab material, but you could say it sort of looks like a horse a little bit. I guess looks like a seahorse. Maybe it's better here, right? You can see better that the hippocampus is essentially a rolled up piece of cortex, right? If you follow it right here, rolled up piece of cortex. And so it does look a little bit like a seahorse. We do have different parts of the hippocampus, and we'll talk about that in just a second, but just to know, where is, like, what's talking to the hippocampus? Well, the first one is what we just talked about, which is the limbic association cortex, which is integrating all this information and connecting to all the different parts of the cerebral cortex. So the limbic association cortex is, of course, going to be talking with the hippocampus. And so it's going to send all types of cognitive information through its parahippocampal gyrus, and that is an extension of the entorhinal cortex. And then the entorhinal cortex, which is going to extend as part of the hippocampus, right? So the hippocampus and the limbic association cortex are continuous with each other. You can see the entorhinal cortex, and that's going to filter the information before it goes into the rest of the hippocampus right here. Okay, the hippocampal neurons are going to process the information, the cognitive information it's getting from the limbic association cortex, and it's going to send it out via the fornix, right? Again, hippocampus formation, then it's going to send out information via the fornix, and then its target is going to be the mammillary body as the primary target, and this is all dealing with memory formation, right? So the hippocampus is really going to be dealing with memory formation overall. But in addition to sending information out via the fornix, it will also send information back to the entorhinal cortex, and as sort of a bidirectional pathway, which we see here. And then it's also going to, via the fornix, also send out information to the limbic association cortex. Okay, so you get information come in from limbic association. Its parahippocampal gyrus extends into the entorhinal cortex, which you could say is the gateway to the hippocampus. That sends the cognitive info from the limbic association cortex, passes it on to the hippocampus, where it is processed, and then you have a bidirectional output back to the entorhinal cortex, but then also output via the fornix into the limbic association, and then, as we will find out, the mammillary bodies. Okay, so additional areas of the limbic system that project to the hippocampus, because it's not just the limbic cortex. We have the amygdala, right? We saw those little masses in front of the hippocampus, and then the septal nuclei of the basal forebrain, as we'll find out. And then those are going to link directly to those are linked directly to the hypothalamus and then the autonomic nervous system brainstem centers. But these two are both going to send information to the hippocampus. And so the result of our fear center and the septal nuclei is you're going to link emotions and motivational states to declarative memory formation, which is the main purpose of the hippocampus. All right, then you also have monoaminergic fibers from the reticular formation of the brainstem. That's going to send serotonin, dopamine, and neuroepinephrine directly to the hippocampus. It's going to alter mood and adds emotion to the memory formation that the hippocampus is doing. All right, so we started to talk about this. So we have 3 different cellular areas of the hippocampus, right? So if our hippocampus is this folded up region right here, this is an entorhinal cortex. Then within the the seahorse that is the hippocampus, we have three different regions. The dentate gyrus, which you can see here, and then Ammon's horn, which is located right here, and then the subiculum right there, subiculum right there. Okay, so you'd say this is kind of like 1, 2, and 3, but information entering the hippocampus comes from the entorhinal cortex via myelinated fibers close to the cortex surface. These fibers are going to perforate the hippocampus, and this perforation from the entorhinal cortex into the hippocampus is called the perforant pathway, but most of the information ends up in this dentate gyrus, which we can see here at the end of it, at the end of the hippocampus. It gets processed by interneurons in the dentate gyrus, and then the dentate gyrus sends additional information to additional projection centers that are located in Ammon's horn, so CA1, CA2, CA3, and then the subiculum, which we see right here. Okay, and then after you have had information come in through the perforant pathway into the hippocampus, we know, of course, it's going to be sent out via the fornix. Okay, but there's this idea that the internal circuitry, the intrinsic circuitry, the interneuron circuitry of the hippocampus is almost all excitatory overall. All right, so then more on the output of the hippocampus, we have the fornix, which we saw was that like tail that kind of wrapped around it, ended in the mammillary body. And so, sorry, I said earlier the septal nuclei of the basal ganglia. I meant septal nuclei of the orbital frontal cortex, which is up here. But you have basically, from the fornix, you have these CA fibers, and those are coming from right here, and or of Ammon's horn, and those are going to go out and out through the fornix into the septal area of the orbital frontal cortex, which is part of the frontal lobe, and you can kind of see that here. So body of the hippocampus comes up via the fornix, and then it's going to extend through this commissure into the orbital frontal cortex. And then you also have subicular fibers, right? This is our other area of the, we have the subiculum down here, and these subiculum fibers are going to go through the fornix, and they're going to go to the mammillary nuclei of the hippocampus via post-commissure fibers. So here's the commissure, and it's going to pass that and go down to the mammillary body. All right, and so this is really the important pathway that we care about, which is the sort of the main output pathway of the fornix is our subicular axons coming from the subiculum through the fornix, and it's going to go to those mammillary bodies. And so the pathway that we have here, this diagram's not the best, I apologize, but you have the hippocampus, and then you have output via the fornix, these subicular subicular fibers, and then it's going to go to the mammillary body, and then that's mammillary bodies embedded in the hypothalamus. And this sort of pathway is important for forming declarative memories, so fact-based memories. And then the mammillary bodies are going to project to the anterior nucleus of the thalamus. So right here, project to the thalamus from the mammillary bodies, and then from the thalamus to from the thalamus to different part of the limbic system, which we already talked about, which is the cingulate cortex. And we know that the cingulate cortex is part of the limbic association cortex, right? So then if you're in the limbic association cortex, in this case the cingulate cortex, well, we know that will connect back to the basal ganglia, and so the final step is to the basal ganglia. So we have through the fornix, the subicular axons, they go to the mammillary bodies embedded in the hypothalamus. This is for forming declarative memories. The mammillary bodies project back to the thalamus or relay station, the anterior nuclei, and then from the thalamus to the cingulate cortex, and then from the cingulate cortex, aka this limbic association cortex, you go back to the hypothalamus via the entorhinal cortex. All right, so it's a circuit. All right, and then finally, what's the clinical relevance of the hypothalamus? This is all about hypothalamus. I'm so sorry. Hypothalamus. This is a typo. This is hippocampus. My goodness. memory formation. So we talked about forming declarative memories. We also have a disorder called Korsakoff's psychosis, which is associated with thiamine deficiency. And so basically you have degeneration of the medial diencephalon. Remember, we say the diencephalon for thalamus, hypothalamus, that region. And so it includes the mammillary bodies and the pathway between the thalamus and the mammillary bodies. And basically, because the mammillary bodies are involved in the formation of these facts and events based memories, if you have Korsakoff psychosis, you have damage to the mammillary body pathway that the hippocampus is all about, so you can't form new memories. All right, and then finally we have transient global anemia or amnesia, and so these are lesions of part of Ammon's horn, CA1 of the hippocampus, and you have transient loss of autobiographical memories of like the long ago past. So long-term memories get lost if you have damage to Ammon's horn. All right, so then next up was, the amygdala, and just a reminder, the amygdala was these little guys right here, and they're embedded in cortex. So the amygdala is also called the aversion center. It's derived from telencephalon. So back to some embryology, and it's positioned anterior to the hippocampus, inside of the medial temporal lobe. The cortex that it's surrounded by is called piriform cortex, but the amygdala is right in front of the hippocampus. All right, and then the bump that is made from the amygdala being surrounded by this piriform cortex is called the uncus. All right, so before we get to that, there's sort of like, I put this out of order, but what's the function of the amygdala? It's serving as the aversion center, and actually very quickly, just more on where it's located. So right here, right here in front of the hippocampus. So the amygdala's function is as an aversion center. It analyzes environmental clues signifying danger. It influences learning and memory, especially under circumstances eliciting a strong emotional response. This is considered a survival mechanism. Stimulation of the amygdala induces attention related to the induction of fear, defense mechanisms, and then fight or flight for sympathetic autonomic nervous system. And then the amygdala, interestingly, is activated when we see fear on faces. We partially know this because of a disease called Urbach-Wiethe disease, where the amygdala has degenerated bilaterally, and so the function of seeing fear on faces is no longer there, and so that sort of generally tells us that if you have destruction of the amygdala, you lose significance assigned to threatening objects, right? Whereas a fearful face would be considered, good sign to have fear yourself. And then neurotransmitter imbalance in the amygdala is thought to underlie anxiety and panic disorders. And we'll come back to the syndrome. But all right, so basically our aversion center. And the question is, how exactly is it getting its information? We have the afferents are sort of olfactory, right? So smell. Then we have the solitary nucleus of the medulla, part of the brainstem, and that's going to give us information about the organs of the body. And the idea is that we want to be able to control the autonomic nervous system, sands and pans, right, with and integrate its functions with emotion. And then the limbic association cortex, which we're familiar with, takes in cognitive information from the rest of the cortex, and then typically it will send specifically visual information is the important one, right, for understanding those fearful faces. Then we have monoaminergic fibers from the reticular formation in the brainstem, and they provide serotonin, dopamine, neuroepinephrine information, which is important for mood. Then information processed in the amygdala, again, this smell, which is important for eating, and then reproduction, and then information related to the autonomic nervous system and viscera being our organs, and then cognitive information. That's what all of this is saying. In terms of its outputs, basically you have a lot of connections to the hypothalamus, which we'll talk about in a little bit, but for controlling feeding, thirst, reproduction, endocrine functions. It also sends projections to the autonomic nuclei of the brain and spinal cord for pans and sands, and then information to the hippocampus to allow emotions to influence memory formation. Then you also have reciprocal connections with the dorsal medial thalamus and then the limbic association cortex. And then the idea is that you're allowing for cognitive and sensory information about the environment to be integrated into like a fear response and memory formation overall. So quick syndrome, the Culver-Bucy syndrome is bilateral damage to medial temporal lobes including the amygdala, which is embedded in that region. You can have visual agnosia, so object not recognized by sight, right? Or fear in faces type of issue, don't assign value to it if it's damaged. Hyper-orality, tendency to examine objects by mouth. Hyper-metamorphosis, intense examine of all objects in the immediate surrounding, or overreaction to visual stimuli. Placidity, failure to show response to fear, anger. Hyperphagia, eating excessive amounts of anything. Hypersexuality, and then amnesia, dementia, and dysphagia. All right, and then finally we have the last part. So we've talked about the limbic association cortex, which is integrating information to help shape cognitive emotional response, the hippocampus, memory formation, the amygdala, our fear aversion center, and then finally we have our gratification center, which is our basal forebrain. So this is located right here, and this would be located like in this region right here that I'm trying to zoom in on. But unfortunately, the software doesn't actually label that, which is a little annoying, but we do our best. So this is the ventral area between the orbital frontal cortex, right here, and then the hypothalamus, which we know right here. So this is the basal forebrain right here. It's also located right here. And it was previously called substantia innominata, unnamed substance, but now we just call it basal forebrain, or we could also call it interior perforated substance, because it has a lot of blood vessels that enter it rostral to the optic chiasm, and you can kind of see those blood vessels. Basal forebrain nuclei, there's two of them, so septal nuclei and nucleus basalis. Septal nuclei, we had sort of defined that earlier as part of the orbital frontal cortex, which I think maybe is fair because it's all like part of this region. Anyways, if we consider it part of the basal forebrain, we have the septal nuclei, and then we also have nucleus basalis. And then, again, this is dealing with the function of the basal forebrain is the gratification center. If you stimulate it, you get pleasure. So what information is it taking in? Well, it's taking in information from other parts of the limbic system, hippocampus, amygdala, association cortex, olfactory axons, and then reticular formation of the brainstem. And then it's outputting to the hippocampus to strengthen memory formation and retrieval. And then, again, this is all associated with gratification. And then nucleus basalis projects to the cerebral cortex overall. And then we also have, adjacent to this, the nucleus accumbens. And this is for regulation of behavior associated with addiction. So it functions with the basal forebrain in mediating behavior associated with pleasure and reward. All right, so that's it for this particular lecture. It's a lot of information. We'll take a look at the next one, and then we're done with NeuroLabs. All right, so here we have the hypothalamus. This is lecture 75, review for lab 10. And so we're just going to talk about the functions of the hypothalamus, and it's our master regulator of basic body functions, talk about some of the anatomy, then talk about its control of the autonomic nervous system, body temperature, thirst, feeding, biorhythms, sleep, reproduction, memory, control of aggressive behavior. But it's a very small region, and it's only, we're told, 4 grams. So really not much to it, but it is the master regulator. So we know that it has some very strong association with the pituitary gland overall, and the hormones that the hypothalamus releases are further released by the pituitary, or it stimulates pituitary to release hormones. So what is the function of the hypothalamus? Well, hypo means below, so we have the thalamus and we have hypothalamus, but it's integrating viscerosensory information, which is organ information, right? Viscera, organ, in order to maintain homeostasis. It's going to control our hormone system, or aka our endocrine system, and as well as the autonomic systems. But every organ in the body is influenced by this tiny region right here, the hippocampus. It is regulating, as we mentioned, basic body functions, so fluid levels, food ingestion, electrolyte balance, energy metabolism, hormone balance, and reproduction. The anatomy is hopefully relatively straightforward, but we divide it into 3 regions. We have the anterior region, which is the contains the optic chiasm, or bounded by it, and then the middle region bounded by the infundibulum, and then the posterior region bounded by the mammillary bodies. The fornix, which we had talked about a few minutes ago, is the output tract of the hippocampus, but it actually runs through the hypothalamus on its way to the mammillary bodies that are at the end of the fornix. The medial and lateral areas contain multiple cell groups that are functionally distinct, and so basically all of the connections between the different nuclei of the hypothalamus allows for sort of a functional integration, such that you could, like, say, have an integration of, a motivation to feed that's coupled with supporting autonomic functions, like maybe you should increase your saliva production, something like that, right? Sans and pons with desire to eat. All right, then we have a whole bunch of different nuclei, and to be honest, I feel like practice in lab is the best way to actually go about memorizing these, so please don't have this be your only resource. We have pre-optic area, which you can see here. This is the anterior division that we're dealing with right here. We also have the suprachiasmatic nuclei, which you can see here. Then we have the supraoptic nuclei, which we see here. And then we have the optic chiasm bounding the bottom of the anterior division, right here it is, and then optic tract right there. Then we have the paraventricular nuclei, which are located right here, adjacent to the ventricle, paraventricular. Then we have the middle division right here, and so we have the arcuate nucleus, and that's located right here at the bottom. We have the ventral medial nucleus, which is located right here. We have the dorsal medial nucleus, which is located right here. And then we have the posterior division, which is bounded by the mammillary bodies. And so we have the mammillary bodies right here, and we have the posterior nucleus right there, and then we have the tuberomammillary nucleus adjacent to the mammillary bodies. All right, so that's not too bad. It's a relatively, as we said, very small area, but it's our master regulator. So we're just gonna start talking about the things that it regulates. And the first off is the autonomic nervous system, which is parasympathetic and sympathetic autonomic nervous system. So rest and digest, fight or flight. But hypothalamic stimulation evokes a variety of autonomic responses. We have a change in heart rate, blood pressure, bladder contraction, changing gut motility, right? Piloerection, which is your hair standing on end. The afferent input about sort of the organs is going to come from a variety of different sources. We were told about the circumventricular organs, which are these sensory regions adjacent to the third and fourth ventricle. We weren't told anything about them in class other than the fact that they just exist. And then the most important one in terms of input into the hypothalamus is something we'd already mentioned a little bit, but this is the solitary nucleus, which we abbreviate as NTS. Why NTS? I think it's for the Latin version of it, but solitary nucleus is NTS. It's located in the medulla, so the brainstem. It collects afferent information from the cardiopulmonary system and the abdominal viscera. So visceral information overall is going to come into our hypothalamus. All right, then we have the hypothalamus is regulating the autonomic nervous system, sans and pons, by projecting to preganglionic autonomic nuclei in the brainstem and the spinal cord, and it's going to do this by sending these axons down this tegmentum along the posterior part of the brainstem. But the examples of its targets, so the dorsal motor nucleus of the vagus nerve, which is located in the medulla for the parasympathetic nervous system, and then the intermediolateral cell column of the spinal cord overall. These axons are going to descend, as we said, through the tegmentum, so the dorsolateral tegmentum of the midbrain pons medulla, again, here, tegmentum. And here we have this diagram as well, but you see we have the anterior part of the hypothalamus and then posterior part of the hypothalamus, and then we have descending innervation to our vagus nerve, and then different cranial nerves to our intermediolateral cell column for the sympathetic nervous system located in the lateral horn. All right, so what if we have damage to descending thalamus tracts? You have something called the Wallenborg lesion of the medulla, which is exactly that, damage to these descending tracts in the tegmentum. And this is going to produce something called Horner syndrome. And this is, problems with the sympathetic nerve supply to one side of the face. You'll have meiosis, constricted pupils, ptosis, droopy eyelid, and then anhidrosis, failure to sweat. But generally, if we were to say try and break down the functions of the hypothalamus based on region, we would say that the anterior part for autonomic nervous system is going to do pans, and then the posterior part is going to do sans. All right, so pans in the front, sans in the back. All right, but not too bad. It's taking in organ information, and then it's outputting via the tegmentum. Pans in the front, sans in the back. All right, So then we have control over body temperature. And so again, we're looking at anterior hypothalamus versus posterior hypothalamus. And so with temperature, like what can you do? You can lower temperature or you can raise temperature, and it's a balance between the two to maintain a constant temperature, right? So the hypothalamus can control body temperature. It can lower temperature by stimulating the anterior thalamus. It's going to cause surface vasodilation in the skin. You can conserve temperature, like maintain body heat, or you can raise it by stimulating the posterior thalamus. It's going to cause shivering, vasoconstriction in the skin, and basically keep heat from leaving your body, and then piloerection or hair cells standing up. All right, then we have the hypothalamic control of thirst. And so thirst, you can imagine, has to do with how much fluid you have in your body. So it's regulated, understandably, by volume in your blood, as well as tissue osmolarity, so concentration of ions in your tissues. And then, how can you sense that? Well, we have volume receptors that are located in the heart and different, the great vessels. And that information is sent to the solitary nucleus in the medulla, and then from there it goes up to the hypothalamus. We have two nuclei from the anterior division that are important, SON and then PVN. So SON is our, let's hope this loads, supraoptic nuclei, so right here, right above the optic tract, and then PVN is paraventricular. So this nuclei and this nuclei are going to be important for regulating thirst. And you have these two nuclei have oxytocin producing neurons and vasopressin producing neurons. It's the vasopressin that's really going to make a difference here, because vasopressin is going to press on those vasos, right, the vessels, and it's also called antidiuretic hormone, so you can think, retain fluid potentially. So we have these vasopressin neurons in the supraoptic nucleus and paraventricular nucleus of the anterior hypothalamus are going to contain additional sensors that detect sodium levels and respond to changes in osmolality, but you also have information coming from the solitary nucleus, from volume receptors located in the heart and the great vessels, and these vasopressin neurons are active by default to maintain a basal level of circulating vasopressin, but during dehydration, you're going to say, produce more antidiuretic hormone, don't pee as much, produce more vasopressin, all right? But the supraoptic nuclei and paraventricular nuclei, which are located here and here, are going to be producing this vasopressin. They also produce oxytocin, but we care about vasopressin here for the control of thirst. All right, then we had hypothalamic control of feeding behavior, and unfortunately for this, we know about it due to some poor rats who got very thin and very obese. And so we have in the mid-hypothalamus region, we have the following nuclei, arcuate nucleus, ventral medial nucleus, and lateral hypothalamic nucleus. So we said we had the anterior section, middle section, posterior section. So we have middle section is going to be dealing with, these guys are going to be dealing with hunger, and then the lateral hypothalamic area, which is here, interestingly enough. And so we have in this infundibulum region, our two nuclei we care about are arcuate, and then ventral medial, and then the lateral region of the hypothalamus are going to help with feeding behavior. And so if you have destruction of the ventral medial hypothalamic region, this ventral medial nucleus, you're going to cause hyperphagia, which is increased feeding. So the ventral medial nucleus is going to house the satiety center, which tells you you're full. So if you destroy the center that says you're full, then you're going to keep eating. Then you have the lateral hypothalamic area, which is going to produce asphasia, which is dangerously decreased feeding. And so this lateral hypothalamic area is our feeding center overall. And so here we have lesion to the lateral hypothalamus, destroying feeding center, you get a very thin mouse. Destroying the ventral medial hypothalamus, you're going to have a very fat mouse because they won't stop eating. Okay, and waiting for this to load. There we go. So then just more on starting and stopping eating. We have the arcuate nucleus is going to help with starting and stopping eating by sensing leptin levels, and the arcuate nucleus is going to stimulate the feeding center when it sees that leptin levels are low. So the feeding center in the lateral hypothalamus. Then to stop feeding, you're going to have GI signals of gut distension. You'll also have an increase in leptin that the arcuate nucleus is going to see. But gut distension via vagus, via solitary nucleus, is going to synapse with our arcuate nucleus, and then the result is you're going to release peptides that inhibit the feeding center of the lateral hypothalamus. Okay, but leptin levels, if you have low leptin, then you're going to say, hey, I want to stimulate feeding, and the arcuate nucleus is going to send out some peptides to the feeding center in the lateral hypothalamic area, and then you are going to, if you have too much leptin, you're going to do the opposite, decrease feeding, because leptin comes from our fat cells. All right, and then there's just a quick thing on different food intakes. You have different neurotransmitters released in the hypothalamus, and upon ingestion of different foods, and so the thought is that different neurotransmitters are going to be linked to the cravings of certain foods. All right, so then we're almost done. We have control of biorhythms, arousal, and sleep. Control of biorhythms, we have basically biorhythms are, you have your body is on like a circadian rhythm. You have a biorhythm of your body cycle, or like body mechanisms to maintain homeostasis in the body, and this biorhythm is maintained by the suprachiasmatic nucleus, and that's going to receive direct retinal input. So, if you're staring at these computer screens at night, it's going to throw off your biorhythm. Not good for us med students, but the suprachiasmatic nucleus is connected to other hypothalamic nuclei, and its goal is to regulate sleep and arousal. To control arousal, you have something called the ascending arousal system, which are brainstem neuronal circuits that promote arousal by projecting to the thalamus, hypothalamus, which we care about here, basal ganglia, and cerebral cortex. The hypothalamus nuclei that are going to be involved in arousal are the are DM and TM, and this should be, let's see, back here, tubero-mammillary nucleus, and then DM is dorsal medial nucleus, so posterior division and anterior division, and then anterior division, again, has the suprachiasmatic nuclei, which is right here. That's going to be dealing with setting the biological clock rhythm overall. But for waking, we have the hypothalamus nuclei getting information from this ascending arousal system, and these two nuclei, dorsal medial, and then the tubero-mammillary, are going to release histamine, and that's going to be a pro-wake-up neurotransmitter. And then antihistamines are going to inhibit arousal neurons, and they're going to make you sleepy or tired. All right, so then we have the control of sleep itself. So pro-sleep, we have the pre-optic area nucleus, which is activated during the sleep cycle. It's going to inhibit the ascending arousal system and help switch from wakefulness to sleep. And these cells are influenced by basal forebrain cholinergic centers, so the septal nucleus, and then the nucleus basalis. And you may remember that from our the gratification center of the brain, the basal forebrain, the septal nuclei, and the nuclei basalis. So we have sleep associated with the gratification center, which is interesting. But the POA, preoptic area, which you can see here, anterior hypothalamus, is going to inhibit the ascending arousal system, help you get some sleep. And then neurons in the middle hypothalamus and posterior hypothalamus are going to do the opposite, which is the dorsal medial nucleus is going to produce orexin, also called hypocretin, to activate the ascending arousal system to wake you up. And the loss of neurons, the dorsal medial nucleus, is thought to cause narcolepsy because your ascending arousal system isn't being activated because you don't have enough orexin. So orexin neurons are thought to inhibit entry into sleep, REM sleep in particular. All right, just a few more sections. We have thalamic control over reproduction and maternal behavior. So basically the idea is that depending on if you're female or male, you have nuclei of the hypothalamus are sexually dimorphic, meaning different shape based on if you're male versus female, different size really. And you also have gender-specific expression of steroid or hormonal receptors. And of the preoptic area, which we said was involved with stimulating sleep, we have also It is also a center for release of reproductive hormones from the anterior pituitary that will be released from the anterior pituitary. And then you have a connection between this preoptic area with the paraventricular nuclei. And you may remember the paraventricular nuclei, and let's go back. Paraventricular nuclei and the supraoptic nuclei are going to help with the control of thirst by producing vasopressin, but the paraventricular nuclei are also going to, and the supraoptic nuclei are also going to produce oxytocin, which we may remember is called sort of the love hormone, right? So dopamine is going to be released by the preoptic area and increases the effects of paraventricular nucleus released oxytocin, the love hormone. All right, subregions of the ventromedial nucleus, the hypothalamus. They have progesterone receptor expressing cells to regulate sexual behaviors related to mating. Not too much there. And then finally, involvement of memory. We have the mammillary bodies, which we talked about in relationship to the limbic system, them being the target of the hippocampus. We said that there was a circuit between the hippocampus and the mammillary bodies, and this would help form declarative memory, fact-based or event-based memories. But the idea is that the mammillary bodies are actually positioned in the floor of the hypothalamus, but they're not connected to the other hypothalamic nuclei as vastly as the other parts of the hypothalamus are. So we believe that the mammillary bodies are, in fact, a separate entity than the rest of the hypothalamus. All right, finally, control of aggressive behavior. We had this study on mice that they severed the cerebral cortex from the hypothalamus, and they found that this stimulated rage and fear in mice, so poor mice. But if you cut below the hypothalamus, so severed this region from the brainstem, you eliminated the rage behavior. All right, so basically this was meant to show that there's some sort of connection between the limbic system with, our fear and rage, the cortex, the cerebral cortex, and then the hypothalamus and brainstem. So a link between emotional behavior, right, rage and fear, as well as autonomic function, fight or flight. All right, and then finally, just general regional summary. So if you were to look at more rostral to nostril, right, this image should be flipped, that's my bad. I mean, the words are correct, but it's confusing because this way is more rostral and this is more caudal. More rostral medial is associated with contentment, control over the parasympathetic nervous system. If you stimulate rostral medial area, you're going to have an increase in passivity, increase in satiety, decrease in body temperature. And if you stimulate more caudal lateral, then you're going to have an increase in anxiety, increase in aggression, an increase in temperature, increase in fight or flight response. All right, so that's it. That was the two lectures we needed for Lab 10, and good job, guys, and, I hope you're doing okay. All right, bye, guys.